All right. Hi, everyone. Welcome to the second semester bio video I've ever made. Today we're going to be talking about meiosis, spermatogenesis, and oogenesis. So without further ado, um, let's get to it. So the important things to know in regards to meiosis are that the process itself reduces the number of chromosome sets from diploid to haploid. Very, very important. Um, Foundational concept right there. The four haploid cells that are produced from meiosis are completely genetically different. So those four haploid cells are genetically different. And genetic variation in gametes arises from the independent assortment of chromosomes, crossing over, and random fertilization. So when it comes to diploid, the somatic cells, they divide by mitosis, they contain 23 pairs of chromosomes, a maternal set and a paternal set. Those are diploid. Our germ cells, however, uh, only contain a single set of chromosomes. So those gametes are haploid. And so that's what causes, fertilization, the union of those two gametes. Let's go on to the steps of meiosis. Once again, very, very quickly, I'm just going to list off the important points. Interphase. Chromosomes are duplicating during interphase, also called the S phase, yielding those sister chromatids. That's the main important part of interphase, is that we are yielding these sister chromatids. Then we got meiosis, after interphase, where overall diploid cells will divide twice, meiosis 1 in meiosis 2, and yield a total of 4 haploid daughter cells. Those products are pretty important. So we're going to start with meiosis 1, because remember I said that they divide twice through meiosis 1 and meiosis 2. We start with meiosis 1. And the goal of meiosis 1 is the separation of homologous chromosomes. So in prophase 1, which is the first step of meiosis 1, we have the centrosomes moving to opposite poles of the cell, the spindle fibers forming and crossing over occurs. Crossing over occurs in prophase 1. Then during metaphase 1, pairs of homologous chromosomes will attach to the metaphase plate. Remember we learned about mitosis. This is a bit similar, if not almost exactly similar. During anaphase 1, those homologous chromosomes separate and then they move towards opposite poles. The sister chromatids remain paired. And then telophase 1 and cytokinesis, the cytoplasm, of course, divides, and then the cleavage furrow forms. In meiosis 2, which is the next step, our goal is not, instead of separating the homologous chromosomes, we're separating the sister chromatids. So in prophase 2, the spindle forms, of course, very similar to prophase 1. Metaphase 2, the chromosomes attach to the metaphase plate, and because we already had crossing over already happened in prophase 1, the two sister chromatids are not identical by metaphase 2. In anaphase 2, sister chromatids will separate and move towards opposite poles, just like in anaphase 1, except we're talking about sister chromatids this time. And then telophase 2, in cytokinesis, the nuclei form, chromosomes begin to decondense, and cytokinesis occurs with the formation of the cleavage furrow. What's really important for you guys to know is how genetic variation arises in sexual cycles. And there are 3 ways that we get that genetic variation. It's that independent assortment of chromosomes that we were talking about during metaphase 1, crossing over in prophase 1, and random fertilization, which means you could theoretically have a baby with anyone, and that produces a whole lot of genetic variance in itself. All right, now moving on to spermatogenesis, which is the formation and development of sperm. So spermatogenesis occurs in the seminiferous tubules contained within the testes, and millions of sperm are produced each day. So each takes about 7 weeks to develop, but in comparison to females, where we're born with all of the eggs we're going to ever have in our life, like males are consistently producing sperm throughout their life, so through adolescence and adulthood. And then the steps of spermatogenesis are as follows. First, we have our primordial germ cells. These are located in the embryonic testes. Second, after the primordial germ cells, we have the spermatogonial stem cell. So we go germ cell, then stem cell, and that is located in the seminiferous tubes of an adult. And then step 3, we have that spermatogonial stem cell turns into a spermatogonia, which is a committed cell. Once that spermatogonial stem cell converts into a spermatogonium, it is set. It is 100% going to become a sperm cell. And then 4, we have our spermatogenesis process where we form a primary and secondary sperm cell. Primary and secondary spermatocyte to be more precise. And then step 5, we have the conversion of the primary spermatocyte to yield 4 spermatids through meiosis 1 and meiosis 2. And then finally, we have our sperm upon the completion of meiosis 2, where the sperm travels through the lumen of the seminiferous tubules to the epididymis and then eventually out. I do suspect that you guys will have to know the journey of sperm, and this is as follows. The testes produce sperm in the highly coiled seminiferous tubules. Then the sperm travels through the epididymis and complete maturation. Step 3, during ejaculation, sperm are propelled via the muscular vas deferens. Step 4, the vas deferens joins the duct from the seminal vesicle, and this becomes the ejaculatory duct. And then step 5, the ejaculatory duct joins the urinary duct, and this becomes the urethra, and eventually sperm exits the cell. So in relation to remembering the pathway of sperm, I like to use an acronym called 7UP. I use this while studying for my MCAT, and the 7UP acronym stands for seminiferous tubule, epididymis, vas deferens, ejaculatory duct, nothing, just because it sounds 7UP sounds better than sieve up. And then the U stands for urethra and the P for penis. So maybe a little test-taking strategy for you guys there. Great, and now we have oogenesis, which is the formation and development of oocytes. This occurs in the ovaries. So oogenesis is interesting because, you know, as I said before, girls are born with all of the eggs that we're ever going to have in our entire life. We're not actively producing them throughout our lives like males are. So oogenesis actually starts when you're an embryo. And as the embryo is forming, primordial germ cell forms in the, many primordial germ cells form in the ovaries, and these turn into oogoniums. And then step 3, primary oocytes form within a follicle. And then what happens after this is a developmental arrest in the prophase of meiosis 1 for these primary oocytes. And these do not continue to develop until we hit puberty. So, all right, now that you've hit puberty, it's time for those eggs to develop. Step 4 of oogenesis begins with adulthood with follicle stimulating hormone, the anterior pituitary, stimulating a few follicles to resume growth and development. Step 5, 1 follicle matures each month and completes meiosis 1, secondary oocyte. That's what ovulation is. So that like every month, one of those follicles matures and becomes a secondary oocyte. Step 6, the secondary oocyte arrests at metaphase 2 of meiosis 2. And if you don't get pregnant, if there is no fertilization of that egg, that secondary oocyte will remain arrested in metaphase of meiosis 2, and you'll end up just bleeding that egg out with your menstruation. But if you do manage to get that egg fertilized, the fusion of the sperm and that secondary oocyte triggers the completion of meiosis 2 for the oocyte, and boom, you're pregnant. Yay. And after ovulation, actually, the follicle that the primary oocyte develops in whenever you are an embryo, that follicle ruptures and develops into the corpus luteum. If the egg is fertilized, that corpus luteum secretes estradiol and progesterone, which maintains the uterus lining during pregnancy. And if it's not fertilized, then the corpus luteum degenerates and a new follicle matures during the next cycle. Now, the journey of an oocyte is not nearly as complex as the journey of a spermatocyte, in my opinion, but that also may just be because I'm a bit more familiar with this, because I have a uterus. But the journey of an oocyte is as follows. Step one, the ovaries, are filled with follicles containing oocytes and supporting cells. Once you ovulate, only one oocyte is released into the body's cavity. Cilia on the oviduct collect the oocyte and move it towards the uterus, which travels through the fallopian tubes and whatnot. Sperm meets the oocyte in the oviduct, which is a part of the fallopian tubes, and fusion results in fertilization. Then the fertilized egg enters the uterus and implants in the endometrium. If you don't get pregnant, the only step that's different is, sperm does not meet the oocyte. The oocyte goes through the oviduct, through the uterus, and out of you. All right, that's all I was going to go over today. Hopefully, I'll be able to do more videos like this in the future as time permits. I hope this was helpful. Bye, guys.